wetlands, and Scott Binger is going to talk about Carolina Bays.
Yeah, hi everyone. So I'm Scott Binger. I'm a PhD student here with the Kennedy Center. And today I really want to talk to you about flora and fauna as indicators of habitat quality in these Carolina Bay wetlands. But before I do any of that, I do want to acknowledge all the folks who support this, all the people who make this happen. I want to thank Crystal so much. She's done so much to make this project happen. Keegan has done so much work to make this project happen. I'm using a lot of data from LL or Chick Gaddy, who is very gracious to share and be involved collaboratively with me. This research is funded by the EPA and has received support from so many wonderful partners who have kind of given me, whether it's land access, different kinds of support. Really greatly appreciate that. So kind of to kick off this talk, I want to talk about the importance of isolated wetlands. So wetlands provide a lot of very valuable ecosystem services. This could be includes like flood mitigation, water quality improvement, carbon sequestration, and also wetlands are great habitat for a lot of flora and fauna. But isolated wetlands in particular, because they're not connected to surface waters of the United States, as defined by the Supreme Court, are not protected under the Clean Water Act. So that kind of shifts the burden of protection on these sorts of wetlands to the landowners that own them, the nonprofits that work with them, conservation groups that are involved with really trying to conserve this habitat. So that takes me to Carolina Bays. Carolina Bays are elliptical depression wetlands. These are often ephemeral, so seasonally drying and isolated. And Carolina Bays have faced extensive historical disturbance, including a lot of ditching, draining the water out of these wetlands, as well as conversion to agricultural use. This map just kind of shows where Carolina Bays are distributed throughout the coastal plain. And you can see right up at the top, it extends all the way up into Delaware and just barely into Florida. But you can see South Carolina is kind of the ideal state to study them in because we just have all the way through the coastal plain, this concentration of these wetlands. So kind of pivoting from the habitat type to thinking about the conservation and restoration. What's a challenge that really dominates conservation and restoration efforts? Prioritizing our limited resources. Researchers and professionals in natural resource fields are really constantly pressed to find ways to make more happen with less money, less time, and they don't have often legal protections to kind of work on to really conserve habitat. So the decision makers that are involved in all of this need tools that they can use to assist with prioritizing which wetlands we're going to work on protecting, restoring, and really minimize the amount of time spent out there assessing them and spend more time kind of moving forward with maybe the challenging logistics that go into all of this, as again, I'm sure so many people in the room are fully aware of. And one set of tools that can really help us out a lot are indices of biotic integrity. So these can be used in rapid assessment of habitats, and sort of the short version of what this framework does is that it's going to use aspects of biotic communities to indicate habitat quality. Sometimes that can be the presence or absence of particular species. It could be the species diversity. It can be all sorts of different things kind of end up being used within this framework. And there's a lot of different taxa that are used for these assessments. So plants and macroinvertebrates have been kind of the more extensive IBI species, and they're often really strong indicators. They're very tightly tied to environmental conditions. However, plants and macroinvertebrates are really time and expertise intensive to sample and identify. It often takes a lot to get out there and ID your plants and collect your macroinvertebrates. Now, on the flip side, frogs and birds have had somewhat less extensive use in this IBI framework. Studies have shown that they can be good indicators, but one of the challenges is that a lot of the effects on frogs and birds are indirect. So while a lot of you might have really strong direct effects on plant and macroinvertebrate communities, the effects on these vertebrates are often downstream of those. So it can be a little more challenging to point them out. But the benefit is that there's a lot of community science opportunities. There's a lot of bird data out there. Everyone's putting their data on eBird. And you can also put out audio loggers. These are calling. So there's opportunities as well to collect data in that capacity and not have to have active surveyors there. So kind of pivoting to thinking about the biotic communities of Carolina bays, there haven't been a lot of widespread studies of the community composition of these habitats. And little is known about how those communities then respond to human disturbance. But disturbances to these wetlands could have really strong impacts on regional biodiversity. Again, these are isolated wetlands spread out pretty far. These could be really important patches of habitat. So we really need to understand these processes in order to adequately assess habitat quality. So just a real quick rundown on sort of some of the taxa of focus and how they're influenced. There are direct effects of hydroperiod, elevation, fire, and then flooding and burning cycles tend to favor diverse herbaceous communities of plants within these wetlands. In terms of how invertebrates are influenced, also very strong direct periods of direct effects of hydroperiod, and certainly indirect effects of the vegetation that's there. And seasonally drying bays in particular can favor those invertebrates that have drying resistant life stages, and can also favor invertebrate predators that aren't getting either outcompeted or eaten by fish. Same for frogs and amphibians in general, direct effects of hydroperiod, indirect effects of vegetation, and both ephemeral and herbaceous bays, as opposed to like the swamps, can really favor a lot of frog diversity. Finally, talking about birds, and especially waterfowl, wetland species nest and forage in bays. A lot of Carolina bays are fantastic habitat for wood ducks, but in general, large heterogeneous bays can support a lot of different niches they can have areas of open water, thick vegetation, sand rims. There's a ton of different ways that these Carolina bays can support bird diversity. And at the regional scale, just the presence of Carolina bays has been shown to increase the total diversity, especially wetland and waterfowl species. So there are three major objectives to my work. Objective number one is to determine how the biotic communities of Carolina bays are responding to anthropogenic disturbance. My second objective is to create IBIs using metrics of community composition that are responding most strongly to disturbance. So again, that could be the total diversity, it could be certain species presence and absence. My third objective is to predict how biodiversity of Carolina bays may be affected by changes in future land use. I'm not going to touch much on this in this presentation for the interest of time, but I would love to talk more about how I'm planning to do that and sort of get people's thoughts on that as well. But getting into my methodology and what I'm currently doing. So the site selection process includes 13 South Carolina counties throughout the northern part of the coastal plain. So this study area extends from Marlborough County in the north down to Charleston County in the south, Sumter County to the west, and Horry County to the east. I'll be surveying 70 total sites. 50 of those are on private land and 20 are on public land. And the private land sites were all selected randomly from an inventory of 143 Carolina bays in this region that Chick-Gaddy did put together. And so I'm going to survey 35 sites per year. So I'll be doing this work this year and next year. Big picture overview of my field methods. At each site, for each taxonomic group, determine either the species or family richness and the abundance of whatever species or families are present, families in particular for the invertebrates. There will be either spatial or temporal replication of sampling so that we're making sure that we're covering a little bit more of a representative range. We're going to measure relevant abiotic parameters as well, including weather, water chemistry, soil composition, and also qualitatively go in and assess site level disturbances. Running through these methods kind of piece by piece. So for frog call surveys, which I wrapped up in May, did 3 surveys per wetland between February and May. Again, record presence and abundance of each frog species, collect weather data to compare. And in addition, I also have audio moth loggers deployed for approximately 40 days within that window, and they're recording the first 5 minutes of every hour. So we can also use that data later on. For bird point count surveys, which I am just about to finish, these are morning point count surveys, and we're doing 2 surveys per wetland between May and July. And again, record presence and abundance of each bird species seen or heard. Same thing, get weather covariate data. We're also using the logger data for bird calls as well. And in addition, we are conducting call response surveys for some secretive marsh bird species. Collecting invertebrates, doing 20 sweeps per wetland wherever standing water is present. 2 surveys per wetland, so 1 throughout the summer and then 1 kind of later on in the fall, right up until right before winter. We'll identify the invertebrates that we collect to family, sort them in lab, and dry them and weigh them to estimate the biomass. And then along with that, record water chemistry covariates like pH, salinity, conductivity, and temperature. Finally, for the plant surveys, it'd be 1 survey per wetland between June and September. Use this nested quadrat design. So we're qualifying in a large circle, all trees, in a slightly smaller circle, all shrubs. And within that largest circle, do 4 plots for herbaceous plants as well. After going through and quantifying with those quadrats, we'll also go through and ID all other species found within that wetland. So that can just give us kind of a total inventory of what's present there. In addition, we'll get some soil information, texture using the field method, soil composition using Munsell color charts. And in addition, during this time, we'll also do a qualitative assessment of wetland disturbance. And in each case, just note the location, type, and extent of the disturbances. There's also GIS methods that we're using. We've gone in and marked the wetland boundaries using US topo maps and satellite imagery. We've calculated the NLCD land cover types and fixed buffer widths. And we're also using that information to gather the intensity of land use. And so this uses the land development index, which just uses weighted parameters where the most intense land use types contribute more to the overall score than the least intense ones. The disturbance gradient that I'm using for this IBI development has 5 major components. There's the buffer width, which is the distance to anthropogenic disturbance from the edge of the wetland in each direction. Land use intensity, again, is that calculated intensity, as well as hydrological disturbance, substrate disturbance, and habitat alteration, all 3 of which are qualitative measures that we'll grab during our qualitative assessment. We use all of those to create a total disturbance score that we're going to use to rank these wetlands. I'm going to get real brief with the analytical methods, but there's kind of 4 major steps. Step 1, select reference and stress sites. So reference sites are your lowest 25% disturbed. Your stress sites are your top 25% disturbed. Establish candidate metrics, whatever the metric is, whether that's species diversity, abundance of a certain species. We just want them to be able to distinguish between the reference and the stress sites. That's kind of the first pass that we'll use to select metrics. From there, we'll go in and do some generalized linear models and model either single metrics or combinations of them. And then we can go in and do some model selection with AICC to make sure that we're selecting the best fit without adding too much complexity to these models. All right, I have some very, very early results. These are based on kind of a subset of what I have so far, but I'm excited to share this. So the disturbance gradient that I'm using doesn't include the qualitative parts yet. We just have the quantitative buffer width and land development intensity scores. And I've just gone in and scaled that, and I'm using that as kind of my disturbance gradient. And I'm using along this axis, I'm using a Poisson distributed GLM because these are count data for species richness. And so what I found so far is that with frog communities, there's no significant relationship between species richness and the disturbance gradient. Same for bird communities as well. We found no significant relationship between richness and the disturbance gradient. You can see actually it's kind of we might expect a negative relationship. We see kind of a positive relationship here. So there's some thoughts about selecting particular species that might be really representative of high quality habitat. Then the last is the macroinvertebrate communities. This is a really tiny sample size, so keep that in mind. But again, found no significant relationship between family richness and disturbance gradient. One thing I did want to mention was the amount of different species of each we found. So we found within all of our surveys, we found 14 frog species. We found at least 39 bird species. And then we found, I believe, 26 macroinvertebrate families. So these overall, some of these sites are very, very diverse and really interesting to take a look at. And I'm very excited to spend time finding out what's really contributing to that. So breaking out really early conclusions, these are really complex relationships. It's going to take a lot of thought and time to sort of unravel what's really going on at these levels to be able to, take a snapshot of a biotic community and use that to give us a lot of information. And one thing I really want to make note of is that inherent differences between the sites in the hydro period, some of these are fully flooded all the time. Some of these are never flooded. These in themselves could be really important defining features, and you can't really always tie that to a disturbance per se. But ultimately, the important goal, why I'm doing all of this, is hopefully to be able to use all of this information to really help prioritize conservation and restoration efforts. My hope is that from building this tool, I want people to be able to rapidly be able to come in and make useful decisions about these really critical wetlands. And these can really ensure the best outcomes for wetland organisms, including waterfowl and also just other wetland specialist species. So kind of what I have going ahead is just finishing up this year's field work. I've still got macroinvertebrate and plant surveys to take care of, getting everything put together to get started on next year's field work and analyzing the data collected so far, kind of breaking down that information more than I already have. And with that, thank you very much. Yes.
John, I think I'm the one that's been emailing you on behalf of the Forest Service. I will say that I would love to see hydro period. Obviously, is going to be the largest determining factor here, but I would love to see differences in plant and community diversity and increase or decrease depending on fire return intervals and intensity. Both of those, I think, are just as large as hydro period in these bays. And I would like to get you, I'm leaving, but I would like to get you in touch with our fire management officer. And so then what you could do when we burn any of these bays that you have on your schedule, you can come back and immediately over the next 7 or 8 years. That's what we see as kind of a natural return interval. Trying to monitor that, see the progression.
Yeah. So yeah, this was a comment on sort of keeping an eye on fire and potentially potentially being a little bit more aware of what's going on with the fire in some of these U.S. Forest Service sites. Yeah, I think that would be so awesome. Well, we definitely talked and thought about it and tried to figure out, even going in and just like scoring based on like fire scars and stuff, but having like information that's that good would be massive for this project, and that would be great. Yes.
You said these areas are isolated; they're not connected to anything, and you said some of them are surrounded by ag fields and crop management. Have you noticed a difference in ones that are surrounded by agriculture? Is the habitat different because of runoff or agricultural practices that might border these areas?
Yeah, this question is about differences in the communities based on the presence of agriculture around them. Yeah. And so what I will say is that generally speaking, what I've seen is that oftentimes you can have a wetland that I would say in many ways is a very like nice wetland, and the wetland itself is not very disturbed. Like there may not be any ditching present in the wetland. The tough thing is that what it seems like is that when you got that kind of surrounding area, there's just less connectivity. There's less opportunities for certain species, it seems like, to access. That's what I've tended to find. I found that if there's at least some sort of connection,
Transmission areas or corridor or something.
yeah, if there's some sort of corridor that can connect it, I think it tends to be a little more, it seems like it's a little bit more diverse. Again, birds seem to have a little bit of an easier time saying, oh, it's fine. But when it comes to like frogs and macroinvertebrates, I would say there's definitely a big difference. But in terms of like wetland, I guess in terms of wetland function, I think for the most part, it's just so variable that it's hard for me to necessarily think about like what's coming in on runoff. I bet that's something that we might be able to detect with our water chemistry parameters.
Say from a management standpoint, if bodies are on private property, you might have recommendations to the landowner about not just how to not disturb this area, but also whether to leave intact a migration corridor, to wooded area, because I don't think a lot of thought is given to what happens around these areas that might actually impact what's in the area.
Yeah, I was gonna say, I'm actually coming up very soon. I've got a meeting with one of the private firms I'm working with, and one of the things I was gonna suggest them. I was like, you have these really nice wetlands, establish some connectivity to them. Yeah. All right.
Thank you.
Thank you.
Scott, appreciate it. All right, so we're coming up on lunchtime, so I think we'll take about a 40-minute break, 30-minute break. Before we go, I'd like to get everybody over here and get a picture before I forget about it, like I did last year and or the year before. And so let's get the picture out of the way. I know how everybody loves to get their picture taken, so. Come on, don't be shy. All people in the back. Andrew needs to be in the back.
All right.